Dear viewers, it is a great pleasure to welcome you to our online webinar today. We are very grateful to you for joining us from different parts of the world, and we appreciate your valuable time and interest in this event. Today's webinar is about the fascinating world of AI tools in early pharmaceutical research. My guests today are David Egan, CEO of CoreLife Analytics, and Philipp Kainz, CEO of KML Vision. We look forward to informative conversations and inspiring thoughts on today's topic. We encourage you to actively participate in the webinar, share your thoughts with us, and feel free to ask any questions you may have during the show. We want to make the webinar as interactive and informative as possible. You can use the chat box to participate in the discussion. Thank you again for joining us today. The interview with David and Philipp will begin shortly. Hello, David, and hello, Philipp. Welcome to our interview. It's a great pleasure to me to have you both here today to talk about fascinating machine learning and also artificial intelligence in drug discovery. You are both scientists and CEOs of your own biotech companies, and you're undoubtedly experts in this field, I guess. I look forward to learn more about you and your companies and the collaboration as well. David, could you give us a little insight into your company and tell us how you got excited about machine learning and AI in drug discovery?
Yeah, sure. First of all, thanks, Stefan, for the opportunity to come and join the chat. I'm David Egan, the CEO of CoreLife Analytics, and my background originally was in molecular biology. I didn't have a data science background, but my career turned into a career of using automation for biology. So helping biologists to do automated experiments, mostly for drug discovery. And so as part of that, later in my career, I ended up helping my clients generate large amounts of complex data. And then that's where I started to get into the data science, because I needed to help them analyze this data. And so that turned into the development of a tool with the person Vinan Domta, who became the co-founder of my company. And then it was a classic startup situation. And so now what we do is at CoreLife Analytics is we deliver software to biologists to help them to analyze complex data. And so, of course, one of the methods that we use in this is, machine learning, or as it's more commonly called, artificial intelligence. So that's our interest in AI. It's giving biologists access to tools like AI, to help them find drugs more quickly.
Wonderful and fascinating. Philipp, I'm also curious about your company and your motivation to work in machine learning and AI. Can you give us a brief overview?
Absolutely. Thanks also for having me again on your podcast. I feel like really honored to be here. So I'm a computer scientist by background, and I did my PhD mostly in the field of histological image analysis. And my motivation was that those big images that are acquired through scanning cannot easily be analyzed with standard tools. So I was also interested in generating methods for the more convenient handling and analysis of those image data. And I founded the company together with my co-founder, Michael, in 2016. And we developed the company from a service-driven company to a product-oriented company over the last years. And since a couple of years now, we are developing and operating the ECOSA platform. It's a software as a service that allows people without any coding skills to train their own AI for image analysis. The application spectrum is very broad, from in vitro to histology to electron microscopy, we are pretty agnostic to the applications. And when I met David, we had a mutual overlapping application field, which was the high content analysis. And so this got us really excited in exploring a little more what we can do there, because our companies have so similar visions in terms of, providing the best possible user experience for biologists and enabling them to be faster in their work, to have less stress in their work, and still get everything done in the time pressure that is increasing too.
So it's very interesting to hear that you both are working together, or your companies are working together. Can you tell us or share more about the benefits you see in this collaboration between your companies?
Yeah, I think, one thing is that, we're well connected with a lot of, biologists in biotech companies and pharmaceutical companies, people who already use our software within their workflows. And we're always looking for, different, more advanced technologies to give them access to. And that's one reason we're working with Philipp, is because of their, vast expertise in machine learning and AI, and especially image analysis. That's something we'd be interested in making available to our customers.
Yeah, and vice versa for us. It's, of course, we are a little more upstream when we compare the positioning of data analytics, the part that David is doing, and we are, of course, drawing the line at some point. We don't want to reinvent the wheel. We are relying on data science experts who make sense of these vast amount of features we can generate from images. And David and his team, they're really good at making sense out of this and providing that at a level with integration in the cloud. That's also one very big advantage to the target audience, and being able to provide a commonly usable solution. That is definitely the goal of our collaboration.
Yeah, I see the combination of your expertise and companies could certainly lead to exciting developments, and I think they substantially can support pharmaceutical research. Well, now let's start with our discussion. As mentioned, or as clear, the topic is the benefits of machine learning and AI, or artificial intelligence, for pharmaceutical research. And I think this is extremely relevant, and I'm sure insights, especially your insights and experiences, will provide us with valuable information. So let's start with the discussion. And this leads me to my first question. What are the concrete advantages you see by using machine learning and artificial intelligence for the pharmaceutical industry?
So if I may start with that, I think the way that people are using devices now to generate data is at a level where analytical capabilities at the moment cannot keep up. So when we introduce our methodologies, I'm speaking now for the image analysis, we can generate so much more meaning from this data that is automatically generated, and it makes it much easier to automate bigger screens, bigger amounts of data. And with those tools, you're able to augment the workflows and enhance, the tooling people are using to achieve their goals. And AI is very adaptive to so many different things, and the way we use it is a little different from the way Core Life Analytics is using it. But still, the principles are pretty similar, and it's super nice to see the versatility of this platform in so many different facets.
Mm-hmm. Yeah, I guess from our point of view, when we're looking at the way biologists are working, in pharma and in biotech, a lot of it is about increased accuracy, increased reproducibility, and also speed and efficiency. I mean, if you look at kind of the basic way in that machine learning and AI can work, you take large amounts of data and you use those to build a model that will help you, for example, in a simple way, for example, maybe to differentiate between a disease state and a normal state. Okay, and building those models takes a lot of work and a lot of computational power and a lot of data, and we can talk more about that later. But then once you have that model and you have an accurate model, then you can apply it very quickly. And someone who doesn't have, let's say, massive expertise in data science, when they have access to good models, then they can use those to quickly identify, for example, the molecule that's going to do a certain job. This could be a chemical to cure disease. There's a company in the US called Recursion Pharmaceuticals who are, this is their whole bread and butter, and it's all about this image-based technologies that Philip and I are so involved in, and it's about identifying images that are disease state and images that are normal state, and then finding drugs that drive cells towards the normal state to cure, let's say, a disease. And I think that's one of these are some of the big benefits, and accuracy as well, taking the human bias out of a kind of an analysis, so that you get a more unbiased approach. Now there are, as we'll talk later on, there are some problems because that can arise with these various models, but that's the potential.
Talking about the problems.
AI,
Sorry for interrupting you, David. Talking about the problems, what are the typical challenges or known potential drawbacks you are facing when developing machine learning or AI related tools and software?
I think definitely Philip should start here, since he struggles with it more than we do.
So I think you need to differentiate between two different aspects. Maybe one, when you have a very specific use case and you try to solve that to the best possible extent, and the other would be if you provide a tool that others can use to basically apply it to their specific problem. In the first instance, I think a big challenge is to understand, get all the data in the right quality and quantity to basically build a model that is of relevant performance. Because, of course, you hear all these rumors, more data, better models, but that is not necessarily true. There is a certain saturation curve you need to keep in mind, and depending on how diverse your data is, you will get there into this situation earlier or later. But of course, since we mostly deal with supervised learning, at least in our domain, when we consider image segmentation, for example, the data annotation is still a lot of work. And to actually be able to assess how good a model is definitely one of the biggest challenges, because it's driven by so many different variables. Is your data representative? Did you train your model correctly? The thing with AI or modern deep neural networks is that you cannot really rely that you obtained the best possible model on that data. That's not a fact. Compared to maybe some other machine learning models like support like the machines, where you can kind of mathematically guarantee basically that this is the optimal solution. However, these different techniques on how to include unlabeled data, but you need to assess, okay, what is the noise that you bring into that? So what is the real value of unlabeled data? Because as I said earlier, you can generate so much data, but not all of that is really valuable. And I think, this pooling, annotations, bootstrapping models, that are techniques you can use to evolve, but it is definitely a trial and error.
Yeah, and then, again, from our standpoint, where we're thinking about, the usability, for let's say biologists, it's all about making the process more understandable for them. And and so if we have a tool, let's say, where we're helping them to pull in data and build models, you want to be able to point them towards the things that may be causing problems. You want your software to be, in a way, some sort of decision supportive, okay, so that it can point out to them, oh yeah, maybe this isn't a good idea. Yeah, you can do it, but try and understand the consequences of it. And that's essential, because there's no way we're going to get everybody trained to be experts in this. And so and this is a kind of a progression in every technological field. In the early days, it's something that's used by specialists. In the first days of let's say automobiles and the internal combustion engine, everyone who owned a car had a mechanic who rode beside them, and so when the car broke down, the mechanic would be able to fix it, and off they'd go again. And then eventually it just becomes something standard that anyone can use, and actually you can't even work on your car anymore. You have to take it to the shop or something, because it's just a device, right? And it'll be the same with AI. Is gradually we'll learn how to build tools that will probably include AIs to build other models or whatever. So these are, I think, some of the the challenges.
Yeah. So it's very important to ensure that there's a decisive quality and reliability of these tools.
Yeah, exactly. Yeah. And that people understand, people can easily understand, let's say, the quality of the model that they've built. Okay. And so that they can see a red flag if it's something that's biased or something like this, and that it helps them to validate models that they're building, things like that. Just take them through the process.
I think the validation is a very good point here. We've actually had a interesting project about 2 years ago where we took a look, especially at trustworthy AI. So what elements are actually essential to make a person trust an AI system, even if it's very specific to solving one use case. It's not a general AI. We're not talking about that. But we were thinking, okay, we need to provide, a lot of insights and I don't know, heat maps and attention maps and whatnot. But in the essence, it was not the technical stuff they were interested in learning, but rather how to coexist, with that and what to do with it. That was much more important than having all the tools that every data scientist dreams about, because they can't interpret it on the spot. They would need to have a training phase, and then they could do it, of course, and smart people. But there's just not enough time. So all the tools we are building, they need to kind of reflect validation steps and make it easy to understand where it is good, where it is bad, in terms of performance.
Do you think that these changes and challenges that come with the use of machine learning and AI now in these days will also bring changes to the roles and competencies that are important for medical researchers, physiologists, physicians, pharmacists, and so forth?
I mean, I talked to people like pathologists about this. I've talked to pathologists about this in the past, and it's there is still a lot of resistance to these types of methods in certain areas. Some pathologists still they want to look at slides down a microscope. I mean, there's still some people out there who don't even want to see the slides on a computer screen. They just want to look at them down a microscope. And people, I think, are they're not crazy about black box solutions. And so I think one of the best ways to kind of introduce it into some of these areas is to, again, make it supportive, so that it can help them to, let's say, prioritize certain things. So, for example, if you take something like you want to diagnose a certain cancer, and you have 500 slides, a certain small percentage of them are probably going to be positive or something. Okay, and so it could help them to kind of prioritize the ones that are more likely to be positive to get a good a good then a good kind of analysis from the pathologist who can confirm that. Okay. And then the good thing about these is some of these ways then of doing that, then it helps to improve the annotation and things like this. If there are errors, the pathologist can pick it up, and then that helps to retrain, improve the model. But yeah, I think the big win in many cases is efficiency, being able to do things more quickly, and being able to get through more work. Being able to speed and efficiency.
Yeah, perhaps without losing any quality, because this is of course the high standard that we need to pursue. And as soon as people will learn how to use this technology, the faster they will adapt to putting that into routine. I mean, there's still a big community of early adopters who are exploring different aspects of this technology, and we had many interactions with people from the industry, and they say, hey, I have this use case, I can't solve it with any other tool. And that is perhaps a bit of a disadvantage to actually start using a new technology, because if you don't know what your standard is producing on this data, then how would you evaluate if a new technology is better? So this is definitely something we are trying to avoid in the first place. We try to start with data sets or with use cases where people are familiar with the outcome and how they arrived at this outcome, and whether they can find themselves and identify themselves with AI technology. But that learning curve is definitely going to be there, and they need to understand, okay, this is a tool I can diligently use for that.
Yeah, I get a bit frustrated sometimes at a lot of these kind of the hype about the evils of AI and things like this, and when people, you don't want this kind of faceless or kind of this sort of anonymous or black box making decisions or whatever. But a lot of the problems that are around the ethics and the problems that will come with AI are the same problems that have always been around whenever you have larger organizations that have power. Okay. And whether this was the railway companies in the 19th century, or the oil companies the 20th century, or technology companies, media companies, whatever, whenever you have a lot of power concentrated in one place, bad things happen. And it's the same, it'll be the same with artificial intelligence, because what you'll have is a lot of data being used to contribute to these various models, and then how they're used may or may not be problematic. If let's say you're talking about building models for healthcare and things like this, are the populations representative, things like that. I mean, that's already a problem with drug development. Our drug development is done for rich Western white people, and there's a lot of problems with the lack of ethnic diversity in things like clinical trials and stuff like this. And so a lot of these issues are going to be the same. Who owns the models? Where the data is coming from? Are there is there informed consent and things like this? So it's a lot of these problems are nothing new, and we'll probably do about as good a job of solving them as we have in all these other domains. So it's I think it's same old stuff.
You were mentioning that this transparency needs to be established at some point. How you get there in order to earn the trust of the public is probably also a little depending which regulations are in place, who is actually in charge of trying to make a standard, trying to create verification processes or quality gates for your developed model. There are some certifications that you can already obtain. I don't really know how well accepted this would be, because when you have a certification that is not known to the industry, you don't have to do it, because nobody, or it won't give you a competitive advantage if you have it. So even if that certifies that you're compliant with some process of best practices or whatever, if you wouldn't do the best practices, you wouldn't be able to survive anyhow, because the model wouldn't work and wouldn't be robust. So I think if you follow that, do a good bunch of education actually in the market, and work to your highest standards on your own in your company with your clients, I think you will be able to move that dial a little bit on gaining more trust into this technology.
Are there further ethical concerns and risks associated with these tools? Do you have anything further in mind?
I think, maybe down the road when, things like AIs are used in, other more kind of predictive ways, especially when it's pulling together data from a lot of different sources, when you're talking about almost this kind of minority report type of situation where, for example, when, there's access to a lot of your kind of previous health data, that someone could predict that you're going to be susceptible to a certain disease, and maybe, insurance companies could make decisions based on that maybe you won't like. That is, those are the type of things that are probably some of the scariest aspects of this.
I think it's rather on a globally economical scale. As soon as there's the masses involved into either benefiting or suffering from those decisions, there will definitely be some discussions on how well did you kind of do the QC on your data. What is basically the driver to actually develop something like this that is so powerful that will affect humanity. I don't think we are quite there yet. We have to still solve so many smaller problems at first. And I would be actually very disappointed if that gets out of hand and there would be one company or one entity creating such a monopoly for decision making that would affect public health in the end.
I know that the pharmaceutical companies do worry about some of these technologies, and I think one thing that they worry about is the idea that a company like Google, for example, would be able to develop technology where they could mine a lot of public information very effectively, and that essentially then they would be able to have kind of corner the market in that, and then all the pharma companies would have to go to them to buy access to this model where they could then use to, let's say, identify targets or identify molecules, things like that. That would be that's not too far-fetched a concern. I mean, we already, the standard search tool is that everyone uses is one company. Just one company. And so it's that idea is not so far-fetched. And it's ironically the drive to open source all this scientific information actually only feeds into that fear, because the more of that scientific information that is open source, then the more valuable a tool would be that could efficiently dig through all that information, combine it with all these data from repositories and things like that to come up with a way to, for example, quickly identify targets that are more likely to be useful in a certain disease. Okay.
Mhm.
And if you could, like, I don't know what, like 1 in 50 or maybe even 100 projects that actually starts for drug discovery ever ends up getting to market. What if you could, what if by using, some sort of, AI platform that you could, maybe, make that 4 times more efficient, right?
Yeah.
That would save you a hell of a lot of money.
Yeah, if you have the right information.
People would be banging on your door to get access,
Mhm.
to that. So yeah, so I think a lot of people were Google are, they're very interested in life sciences. We bump into them, a lot, and it's kind of right in their wheelhouse. Huge amounts of information out there. How can we kind of synthesize it all, pull it together to, make better predictions, more accurate predictions?
That's the idea. Coming from the global issues, coming back to more methodological issues, what are pitfalls or sources of error do you have in mind, Philipp, that can occur in the implementation of machine learning and AI for your research projects?
So I think the expectation of when you have access to AI that you can solve it is a big problem, because it's not given that you can solve this problem. As I said, there are so many variables, and if your data doesn't just contain these, in our case, visual cues to discriminate, 2 different disease states, then you cannot really solve it. I mean, you can go higher dimensional and becomes even more opaque, and you can't really interpret what it's doing. And at some point, I'm almost sure that if you go deep enough, basically you will find some difference. If that is causal, I don't know. How can you check that if you're so deep into this high dimensional space that you basically just introduced a model selection bias because you selected the model that just works for you? So it's a self-fulfilling prophecy in the end. And one other thing is that you should always kind of do this in different steps. So if you tend to work hypothesis driven, then of course there is a certain step of getting closer to your answer. You shouldn't start with edge cases, for example. You start with the normal thing, try to, let's say, in a Gaussian curve, get the 67% right, and then take care of the rest, and try to assess and work with validation methods that assert the stability of your method. And as soon as you know, okay, you have a stable model that behaves on this data, you can go and expand on the other data or edge cases. And probably you can't really cover all the cases, but you should at least know what your model is doing in those situations. And there can be, adverse attacking your own AI, just to make sure, okay, what is happening? So try to break it, for example, in your QC. And that goes a little bit, beyond the standard that what means, okay, I validate and I held out test set. That's pretty basic thing you need to do. But going beyond that is definitely required. And sources of error in that sense is just, selection bias for your data, having too much label noise, which means you don't have, in the supervised learning setting, you don't have a consensus on your labels, which is super often the case because the biology is continuous. You cannot say now this is in that state and now this is in that state. So there will always be some wiggle room, as we call it. And if the model is able to catch that to a certain extent, where it's producing a consistent and systematic error, you're in luck. That this is good. Whereas if it produces like random errors, you're not better than a human, and you should always be aiming for these stoic, repetitive tasks to be better than a human.
Yeah, I think again, it's the involvement in humans in the process is the problem. As kind of Philip was kind of talking about there, science has traditionally been kind of hypothesis driven, but unfortunately, hypothesis often becomes dogma. And you know, this really struck home to me in the early days of our company when we would be working with with users and customers initially, and you know the idea was they would do image analysis to generate numeric descriptors of what was going on in cells, and then they would import that into our software, and then they'd be able to visualize these and graphs and things like this so that you could see the differences, clearly the differences between phenotypes. This is cells where you have nothing going on. Here's cells where you have something going on. And very frequently you'd have these cell biologists who would show you two images and they say, yes, you see, these are different because here you can point out the differences in this one, and you don't see as much in that one. And then, okay, well, let's have a look in the data. And then we run them through the image analysis, extract the numbers, and then when we'd actually see sometimes when we'd see these data visualized in a way where it's just spots on a two-dimensional graph, there's no difference. And it was really, you could tell it was just human bias. There was a hypothesis there. If I knock out the function of this protein, it's going to affect, I'm sure it's going to affect this process because I read a paper about that or something. And then of course people see certain images, as Philip said, that are they're not representative, and they're just, and it's not fraud or anything. It's just human nature to want to be right and to want want to see the result that you want to see.
Want to see
and
their own dogma, yeah.
And then that kind of then can feed into things like like building models and things like that, and special annotation. And this is kind of one of the biggest problems is the annotation of the data that's required to build certain models. And so it's removing that kind of bias and things like that, that's critical.
I experienced this scenario a lot, and it was always very difficult, although I had the statistic on my side to convince the elder scientists that their dogma or their hypothesis isn't true. And going a step further, coming to cell painting, which has not been possible without machine learning and AI tools now that we have in place. How does this approach has already improved the efficiency and accuracy of identifying new potential compounds or new drugs? What do you think? Or do you have already experiences?
Yeah, I mean, a lot of our customers are using the cell painting, technique, and it appears from talking to people in the field, there are some things that it's clearly good at. Some things maybe not as good as maybe people would have hoped. A lot of the original kind of hype was, oh, you can use it to figure out the mechanism of action of an unknown molecule, things like this. Things often aren't so easy. Where I see people having the most success with it are probably in things like toxicology, also things what's called lead hopping in small molecule discovery, where you can take a molecule of interest, generate a profile, compare that to other profiles, look at those molecules, and then use that to inform the medicinal chemistry to help you more quickly improve the activity of molecules, or to come up with a very different type of molecule, so that you can make bigger jumps in medicinal chemistry. So these are the things that I think are very useful. But like everything else, the cell painting, often you're depending on having a good repository of data to compare the data that you're generating compared with that data. And again, the old problem of reproducibility, raises its ugly head, and these are definitely pretty serious challenges. Reproducibility across different sites, people using different instruments, things like this. Yep.
I think that the way that you can actually generate a lot of fingerprint data for cells, for objects, for plate level, well level data, it gives you the opportunity to look at different scales at a problem. So you can basically take the meta perspective. You can try to figure out at different levels what's the difference, to pool that data and be able to do a drill down. When there's something odd behaving, or if you do like a plot of your data and you see, okay, there's just one big bunch of dots in the middle. How can you basically add meaningful information to differentiate those dots, to kind of disentangle the biology, which is of course the goal of this, to understand what's happening there. I think the AI or ML methods, they can be used in very different forms. So ones would be if you take cell painting, and there's different approaches how you can do this. I mean, we use a segmentation approach because it's interpretable. You see if the cell was segmented correctly, or if there are parts that are not in line with what you are expecting. I think there's, you can go full black box. You can say, okay, I just want to predict a random number, a random sequence of numbers. It's not really random, but it's a representation of the image of the objects of whatever. And then you try to cluster that in a high dimensional space. But you lose the ability to track what is happening along the way. So when there is, for example, a model trained on the JumpCP pilot data set, for example, we know, okay, there are 2 different cell types in this data set. We know that these compounds are in there, and so on. And then you train your model, you validate your model on this specific data set. And once you go like outside of this lab environment, there is anything else that can happen. So to assess also, okay, what is the potential behavior of the model when it encounters something in the wild that it didn't see before? Does it cluster that or create features for this specific state into one of the compounds? Is it reproducible? Does it happen all the time? If you repeat in training the model, does your second model behave the same way? Then there must be something related to that. But as people are doing technical and biological replicates in the screening process, there's definitely a need for cross-validation in not only cell painting, but in general in AI methods, because you may accidentally select a very representative and good training set, but once you perturbate your entire pool and repeat this again, you may end up with a crappy model, and you need to make sure that this is a consistent quality. So I think that is definitely an effect they will have also on identifying new compounds and drugs.
Mm-hmm. But to be honest, I'm very convinced about cell painting. Not to get me wrong, but on the other hand, all the cell painting data we're looking at these days are relying to a very, very limited number of cell types, which are cell models, and on a very limited number of impact points, I would say. We don't have any live analysis. We have, let's say, about 2 to 3 in the maximum time points. We look at the impact of the compound. Is this sufficient enough to have reliable data?
Yeah, I mean, you're talking about a snapshot in time, and ideally, of course, you would have, concentration, dose, multiple doses of your molecules and multiple times, and maybe even multiple, cell types.
Yeah. Maybe even 3D approaches to look how does this reorganize the cells.
Yeah. But when you look at the types of environments, and again, this is something that we're really tuned into because we're talking to biologists who are at the coalface. Yeah, it's just not practical at the moment.
No.
And so this is that's really it's limited by the ability to do these type of experiments, and most people are still doing them in well-based 384 well, 1536 well plates. So so we're still at this kind of place where you take your U2OSs and you use that as a kind of a target agnostic kind of detector to try and get a some sort of commonly usable profile that can be used across different platforms. So I guess things like microfluidics and things like this, or some of these other technologies, maybe these could be useful. I think maybe as opposed to trying to I think maybe one way to get around this is maybe kind of multi-omics. And as opposed to trying to expand with cell painting across all these different, let's say, concentration times, things like this, maybe, and that could be something that AI could be used for, would be to try and combine these different types of omics technologies and data from different omics technologies. I think that might be more useful. And so image-based kind of cell painting approach, some of these cellular mass spec approaches, obviously RNA-seq for gene expression, things like this. Being able to pull all those together and take a multi-omics approach might be more useful. I don't know.
Yeah, I think so too. I mean, if you consider if you would be able to run different kinds of analysis on the same sample, then the value you gain is much higher than going for different dose response or whatever 3D. And if you just multiply those numbers, you will need a truckload of hard disks to even store image data, let alone transfer and process it. So that is not practical. And I know, Stefan, we had a discussion earlier as well on how valuable is 3D at the moment, and should people go for that? And I always say it depends, because in some ways, of course, it makes sense if you don't see structures when you fix a certain plane, or if you do a projection, you have to go 3D, and it does make sense. And this is biologically motivated just by filling some blanks into a grid search of different variables that can interact with each other. It's probably a brute force approach that is not as expedient. Maybe it is, but it's not as practical in the end to reach your goal. You generate a lot of data, you need a lot of money to process it, and in the end, it was a nice exercise. And what do you do with that afterwards? So are you going to reuse it? Are you going to mine it for different purposes? I mean, that is something our clients are also interested in. Okay, so I generate this data for this study. I have a client. This client wants to, do some additional readouts on that data, or they are interested in, okay, what else are interactions or relationships that can be discovered, and those discoveries can, of course, be accelerated by AI, because many of those patterns humans can't just see. It's not possible to grasp it. And I think that is definitely also a future direction of how you can combine different technologies to enrich the readout in a more meaningful way.
Yeah, well, that brings me to our outlook what are your opinions? What are the future aspects of AI, cell painting, 3D, and the use? Let's figure out in early pharmaceutical research over the next 5 years, not further, I would say.
Mm-hmm.
Yeah, it's a tough question, because predicting the future is stuff for AI and not for us. But
I would guess AI would come up with a suggestions or an idea or a fantasy.
Yeah, absolutely. I mean, I think the outlook would be that once the adoption happens and we are aware of where exactly the value can be generated using those tools diligently and meaningfully, this can tremendously accelerate research in this domain, and at the same time cut down the cost, because you will be able to, if used correctly, detect and determine potential pitfalls in the next but one step that you will hit down the line when, I don't know, bringing a drug to market, for example. And also in terms of, scaling up experiments, scaling up research processes across different sites, standardizing your toolkit. This is all, possible, but I mean, standardizing tools has been around for a long time. You can ensure, if you have the proper QC and QM system in place, that everybody uses the same tooling. But what was lacking back then and what is enabled by AI now is fast improvement in iteration. So if you figure out, okay, your tool, your ruler, for example, is bent, then you probably need something to fix it, or a new ruler. And with AI, with this adaptive thinking, you can basically create your new ruler that fits your new requirements much faster and with less efforts that has been possible before.
Yeah, I think, in the next 5 years, it's really about, again, kind of increasing the efficiency, identifying, let's say, problem molecules or problem compounds, dumping them out at an earlier stage. So that, it's about this, having a higher chance of getting something more quickly through the process. Not having to, having only to, let's say, make 250 molecules as opposed to 3,000 molecules to get to where you want to be. And if you can do that, that's an enormous, benefit. Because all of those things, all those molecules have to be tested in so many, different types of experiments. If you can cut down on, or eliminate, animal use, that would be of enormous benefit. I think that's the type of thing that could be, we could get towards over the course of like the next 5 years or so. Yeah.
Perhaps also the way we work with the AI will definitely influence how fast this adoption will happen. I mean, these generative approaches that are currently hyped in the news in terms of, text generation, NLP, all of that. There are, light tower projects in the life science domain that can generate, protein folds and predict stuff like this. So I think it's in both. So approaching from both ways, coming up with innovative or letting AI produce innovative new structures, and at the same time improve the standard how you measure the effects of those things, will definitely accelerate also in the short term. You don't have to wait for 10 years. The technology is already here.
And this is a good point, Philipp, what you said, that AI will evolve, and also the user interface, how you deal with AI is getting better and better and more efficient for us in the way how humans are interacting with these artificial intelligence tools, so that it is also can become a common use for the common people or the common scientist, that the biologist and the pathologist can use this tool more intuitively as it is already used. I think this is a good try.
If people are interested in having a look at, where all this could eventually lead us, I would recommend a science fiction series by a Scottish writer called Ian M. Banks. And so he's written a whole bunch of books. He died in 2013. But he wrote a bunch of books based around a thing called the culture, and it's where these advanced AIs they're actually placed in interstellar spaceships are called minds, and we've become so advanced that the minds, these AIs, take care of everything, and humans are just free to live their best lives and engage in things like advanced body modification and things like this. So that's I'd recommend the culture series.
They are fun.
Yeah.
Cool.
So this is what happens if you give too much time to people.
Yeah.
There you go. Exactly. Yeah.
Thank you both so much. It was very refreshing, exciting, and wonderful to talk to you today. Thank you so much. Have a good day. And
okay. Thanks a lot, Stefan.
Bye-bye.
Take care.
Thank you, Stefan.
Bye.
Thanks, David.
Thank you for joining us today and staying with us until the end. We hope to have enriched your knowledge and understanding of AI tools in pharmaceutical research, as David and Philipp had their exciting discussion and shed light on the topic with their expertise and experience. Thank you for your active participation and insightful questions. Thank you also for your time and attention. We look forward to welcoming you again soon to our next webinar. Enjoy your evening and take care. See you soon. Bye-bye.
Record